So my presentation topic is gel electrophoresis. We talk about gel electrophoresis. The outline: what is gel electrophoresis, principle and factors affecting standing stock, gel loading die, and these two types of gel electrophoresis and their applications. Electrophoresis, when charged particles move in electric field in a gel medium called gel electrophoresis. Electrophoresis is most commonly used for biomolecule separation such as DNA, RNA, or protein on the basis of their size. This is the gel electrophoresis is current supply anodes, cathodes, and gels, shell casting tray, and porous gels and buffer solutions. You can see here principle: when any charged molecule in the electric field, they move toward the positive or negative pole according to the charge they are having. Proteins do not have any net charge, whereas nucleic acid have a negative charge, so they can move towards the anode when electric field is applied. Father of gel electrophoresis was Ernie Teslius, and he got a Nobel Prize in 1948. The next slide is factors affecting of gel electrophoresis. Electrophoretic velocity depends on inherent factors: how much charge the particles have, what is the molecular weight of the sample, secondary structures like shape, external environment, the pH of solutions, electric field and solution viscosity, and the temperatures affects the gel. And now we note about the gel. As proteins and nucleic acids are electrophoreous moment under the affecting electric field in the gel. Usually the gel is polymerized in the shape of a thin slab and having wells to load sample. Agarose is a polysaccharide extracted from seaweeds and contains many agarose subunits. During solidification, agarose from a network of polymers, and its pore size can be determined by its concentration. Here, when the concentration of the agarose gel is high, then the pore size is small. When the concentration of the agarose is less, then the pore size is large. So this pore size of gel depending on the concentration of agarose. It is usually used to have the concentrations of 0.5 to 2%. Agarose concentration is a higher. Heat agarose with the vapor to prepare gel, and after it's cooled down, it's poured into the tray called casting tray. These gels are not toxic unlike acrylamide gels. Range of separation in agarose gel is higher, but resolving power is low. By varying the concentration of agarose, we can separate 500 to 4000 base pair of DNA. Next, the staining is more important to see your sample movement. Stock prepared and precaution. Ethadium bromide, we use many integration between this base pair of nucleic acid, resulting in a very strong binding. When the ethadium bromide is exposed to ultraviolet light, electrons in the aromatic rings of the ethadium bromide molecule can activate it. The electrons of ethadium molecules activated and produce release energy in the form of light. Here we can see when, because DNA or RNA have, they are colorless. When you standing this ethadium bromide, they produce blue color, and we can see the movement of our samples. So therefore we add ethadium bromide and other chemicals. Stock prepared 10 mg ml upon ml. Working concentration is 0.5 microgram upon ml. Precaution: ethadium bromide is carcinogenic in SOPs of lab and PPE to protect yourself and environment from where you are working. So we use different SOPs. Here we can use gloves while handling the stain and stand gels. So the next, loading the dye and samples. Loading dyes which are used in gel electrophoresis, they have three main rules. The first one, they add weight to the sample, so it gets settled down in the well properly. They provide color and are visible, so it's easy to load the sample. Here you can see that the dyes move at steady rate, like you say, the moment of light with your sample. So we can get estimation about how far DNA fragments have moved in the gel. So like in by these force agarose gel electrophoresis, these chemicals we use. The type of gel electrophoresis is sodium dodecyl sulfate polyacrylamide gel electrophoresis SDS page. The standard test that used to determine the charged molecule, mainly proteins and nucleic acid. History lamellary system of SDS page was first introduced in 1970. This is the structure negative charge like agarose gel electrophoresis, and they supply the electric field and buffer solutions we can use in this SDS page. The second is 2D gel electrophoresis. It is a type of gel electrophoresis where proteins are separated in two dimensions. It is the only method which is available in and is capable of simultaneously separating thousands of proteins. Basis of separations: proteins are separated on the basis of two properties. Firstly, they are separated on the basis of their net charges. Page separates the protein on the basis of their mass. Here, page separates the protein on basis on their mass because protein have no net charge. Protein is neutral. Therefore, they separate by their mass. Using this method, small change in the charge and mass can be detected. Hence, it is not possible that two different proteins will resolve to the same place in both dimensions. And this is the structure of gel 2D gel electrophoresis, low pH and apply electric field. And they are the small size of the protein non-acidic, and the large sizes are more basic in nature. These applications are of the gel electrophoresis. Gel electrophoresis is used in many fields of science, widely used in the biochemistry, forensics, genetics, and molecular biology to determine the purity of protein samples, molecular weight of protein, quantifying proteins, blotting applications. Gel electrophoresis is most commonly used for biomolecule separation such as DNA, RNA, or protein on the basis of their size. So, thank you.